Hello, my name is Fernanda Fermento, and today I'll be talking about phototransduction of rod cells in the retina. The retina contains rods and cones. Rods help our eyes adapt in the dark, and cones helps us see color. Both rods and cones contain discs, connecting stalks, mitochondria, Golgi apparatus, and nuclei. When a particle of light called a photon enters through the pupil and the lens of the eye, eventually hitting the layer of retina in the back of our eyes into the rod cells. This photon hits the cis retinal, causing it to change shape into transretinal, which therefore loses its attraction to the opsin molecule, therefore freeing the binding site of the opsin molecule, therefore allowing the transducin and one of its GDPs to bind to the opsin molecule, therefore changing that GDP into a GTP with an addition of a phosphate, and this GTP attaches itself to one of the alphas of the phosphodiesterase, leaving one more alpha on the phosphodiesterase. Steps 1 through 6 must be repeated one more time for the removal of the second alpha attached to the phosphodiesterase. By the entrance of a particle light, again known as photon, hitting the cis retinal, causing it to change shape, changing it to a transretinal, therefore losing its attraction to the opsin molecule, freeing up the binding site of the opsin molecule, therefore the opsin molecule will bind itself to one of the GDPs of the transducin, changing that GDP to GTP with an addition of a phosphate, and that GDP will attach itself to the second alpha of the phosphodiesterase, freeing up the phosphodiesterase. When no light is present, cGMP-gated sodium channels are open, allowing sodium to enter the cell, releasing neuron transmitters, but with the presence of phosphodiesterase, converting cGMP to GMP, closing the sodium channels, opening the potassium channels, which starts to hyperpolarize the cell.